Hello, and thank you for attending today's webinar, The Evolution of DNA Production from Cells to Chemistry, presented by Elegen and Fierce Biotech. My name is Janelle Karimoto, and I will be your moderator. Before we begin, here are a few housekeeping items. To learn more about our speakers, please visit the speaker bio window. If you accidentally close a window, you can reopen it by clicking its name in the top navigation bar. This webinar is being recorded and will be available on demand within 24 hours. And finally, we will host a Q&A session at the end of the webinar. Feel free to submit your questions at any time using the Submit Questions window. It is now my pleasure to introduce our speakers. Joining us today are Dr. Michael Junkin, Associate Director of Fluidics and Integration at Elegen, and Dr. Brittany Ensman, Senior Product Manager at Elegen. We'll get to the presentation shortly, but first, a bit of background. Synthetic biology has long relied on cell-based cloning to manufacture DNA, where bacteria are transformed with DNA constructs and colonies are screened to isolate a perfect sequence. However, this approach has clear limitations, particularly when producing complex sequences such as long repeats, high GC content, and hairpins. It's also a slow multi-step workflow that carries risk of contamination and endotoxins. Cell-free synthesis addresses these challenges by eliminating cells from the workflow, enabling the rapid production of long, complex sequences with high accuracy. In today's webinar, our speakers will introduce Elegen's compact Gen 2 cell-free DNA production platform, which combines a microfluidic oligonucleotide synthesizer with cell-free cloning technology. You'll learn how the platform leverages machine learning and robotics to fully automate DNA production, delivering NGS-verified linear and plasmid DNA with fast turnaround and at low cost. We'll also cover how this platform can produce more than 10 times more genes, while significantly reducing footprint and capital costs compared to traditional cell-based approaches. At any point during the webinar, please feel free to submit your questions using the Submit Questions window. All right, let's get started. Dr. Junkin, over to you.
Thank you for the introduction. Again, I'm Michael Junkin. I'm the Associate Director of Fluidics and Integration here at Elegen, working primarily on Elegen's microfluidic oligosynthesizer. Since our first launch of our product in early 2023, we have rapidly developed a broad offering of products produced from our proprietary cell-free DNA production platform. Today, we are shipping thousands of genes every week to customers all over the world from our headquarters here in Menlo Park, California. What does that consist of? So we have Infinia linear DNA, so that's fast turn, NGS verified, high accuracy, double-stranded DNA. That's then followed by Infinia IVT ready DNA. So that's ready to use linear templates for mRNA with poly A tail available. And we also offer Infinia plasmid DNA. So that's long and complex plasmids delivered with a fast turnaround time. In our webinar today, we're going to discuss some limitations of conventional DNA manufacturing, and then introduce the core technologies which Elegen is using that enable our cell-free DNA platform to deliver DNA products unlike any other synthetic DNA offering currently on the market. Let's start with some of the limitations of conventional cell-based DNA manufacturing. For decades, gene-linked DNA has been produced by hybridizing oligonucleotides into fragments, then assembling these fragments into plasmids, and then putting those in bacteria. So what does that look like? First, you have to produce small pieces, oligos of DNA, with various methods, assemble those up into larger constructs of DNA. Then you have to put those into bacteria. You have to then grow those bacteria up, plate them out, screen them, do sequencing on these different colonies, and then do colony picking. And then once you've identified your colony of interest, you have to then put this DNA back in solution, scale up that DNA, scale up that bacteria, grow that until you have enough usable material, then break those bacteria apart, and then purify your DNA out of that for downstream usage. And now, while oligo and fragment synthesis has advanced significantly over the years, the error rates that are inherent in this process across a gene-length construct still remain too high for direct usage. So as a result, cells are still needed for this cloning process to isolate and amplify sequence-perfect DNA. And what that amounts to is it adds time, cost, and operational complexity to the manufacturing process overall. So then if we look at some of the pitfalls of bacterial cloning, we have things like we mentioned, long production time. So this long production time essentially slows development time, time to market, and overall slows down innovation. You also have a recombination risk. Again, when working with bacterial cells, you have to put the DNA into a bacteria so you can get undesired changes to the structure and content of your piece of DNA of interest. You can also get limitations with the design space where cytotoxic sequences cannot be made. Again, whatever sequence you're looking at has to be compatible with the bacteria, so that limits what you're able to ultimately manufacture. You also have things like endotoxin or LPS contamination, which reduces the transfection efficiency and cell viability and the safety risk. And even in the best of scenarios, you have to produce a lot of DNA with a lot of endotoxin, and you have to purify that out in order for your DNA to be used downstream. You also have a large physical footprint. So if you think of growing DNA inside a bacteria from an operational perspective, it also requires significant infrastructure. You have things like liquid handlers, sterile tissue culture facilities, fermentation capacity, and a large, highly trained workforce. As a result, scaling, localizing processes to certain areas, or even incrementally improving production is very capital intensive and slow, and can even be a hard limit for certain organizations. Elegen was founded with the singular goal of fundamentally rethinking how DNA is made. So eliminating cells from the production process was the first and most critical step in enabling faster, cleaner, and more scalable DNA manufacturing. So now let's dive into Elegen's cell-free DNA synthesis. At the heart of everything we do at Elegen sits our cell-free DNA production platform. Our second generation of this platform just came online, and it integrates our patented microfluidic oligosynthesis technology with our fully automated cell-free cloning process. This system is compact and is producing thousands of genes every week without ever touching the cell or compromising accuracy or quality. So if we look at what that consists of, we have basically a benchtop microfluidic oligosynthesis platform, which is capable of parallel synthesis of thousands of oligonucleotides in a single run. We have a cell-free gene synthesis platform. So this is a work cell which takes DNA assembly and does it in an automated manner. And then we have cell-free cloning, which is also an automated process producing thousands of full-length sequence-perfect constructs every week. So we contrast cell-based traditional cloning with Elegen's cell-free cloning. We can see that the cell-based cloning is slow. This process can take weeks, in the best of scenarios, even longer if you have to go back to this process again if you have a failure. Contrasted with cell-free cloning, which is fast, Elegen can ship out DNA within 6 to 8 business days. There's also a high risk of recombination. As mentioned, in traditional cell-based cloning, cells are used, so you have recombination risk. And again, with cell-free cloning, no cells, so there's no risk of recombination. There's also the design limitations. Toxic sequences can be handled much better with cell-free cloning because you don't have that limitation of needing to work around what cells are capable of. Again, with cell-based cloning, as mentioned, high risk of endotoxins. With cell-free cloning, there's no cell contaminants, so no endotoxins are present anywhere in the process. And then with cell-based cloning, again, there's large infrastructure. You can think of lab scale, factory scale infrastructure needed just to produce a usable amount of DNA. And with cell-free cloning, you have a very highly miniaturized, very automated, very scalable platform capable of producing high quality DNA. With our cell-free platform, we now produce linear DNA fragments, each individually NGS verified for sequence accuracy and purity. While conventional gene fragments are produced from 3 to 5 kilobase paired lengths with error rates from 1 to 3,000 to typically 1 in 5,000 bases, our gene fragments can have significantly better metrics. So what does that look like? We have Infinia linear DNA. So this is individually NGS verified gene fragments ranging in size from 300 base pairs up to 7,000 base pairs with a 1 in 70,000 per base pair, so very low error rate, and this can ship in 6 to 8 business days. We also offer Infinia IVT ready DNA, so this is also NGS verified IVT ready templates ranging from 1 kb to 5.5 kb in length. And we also offer customers poly A tails up to 130 base pairs in length, and this ships in 10 to 15 business days. And also by demand, we do offer our Infinia plasmid DNA. So this is, again, NGS verified clonal gene synthesis, ranging in size from 300 base pairs up to 15 kb. And this ships, this is plasmid DNA, but it typically ships in 10 to 15 business days, so a quicker turnaround compared to traditional plasmid DNA. So now let's talk about Elegen's DNA innovation and how we're able to offer these products to the market. Our cell-free DNA platform is built from 3 innovative proprietary technologies, the first of which is a miniaturized oligosynthesis platform. That's followed by directed high-specificity DNA assembly, and finally, our cell-free cloning platform. So we'll talk about each of these in more detail in the coming slides, but it's important to understand that while some DNA suppliers may have innovations in 1 or 2 of these areas, Elegen is the only company that has applied innovative methods and technologies throughout the entire end-to-end DNA manufacturing process. If we look at the first part of the process, that's our microfluidic oligosynthesis platform. This is a high throughput synthesis platform with subsetting capabilities. So the core of this is a split pool methodology. With this split pool methodology, we're able to produce thousands of oligonucleotides simultaneously and incorporate multiple orders per run. If you think about conventional split pool methodology, you split and pool your substrate or your beads at each step. But at Elegen, we do that a little bit differently because we're doing this inside microfluidic channels. We have something which is called encoded positional information. And what this does is it enables us to do precise tracking and quality control of each solid support or bead, and thus each oligo at each step throughout the process. So essentially, we track these beads continuously throughout the whole process of building up a linear piece of DNA. We do that on this chip by having different channels. So if you look at these 4 middle channels, these 1, 2, 3, 4 channels, that would be the split part of the process. When the population is split, we add a base or a piece of DNA onto it, and then proceed to the next part where we do the pool. So that's when the beads or the solid supports go into these 2 outer spirals. So we do the split pool over and over again whilst tracking these beads continuously. And one of the other core technologies is we use microfluidic steering. So throughout each part of the DNA synthesis pipeline, the beads must be directed to different areas of the microfluidic chip. So we do that again with microfluidic steering, where we're using optics and fluidics to guide individual beads to specific channels for specific nucleotide addition at the desired point in the building up the piece of DNA. If we look on the upper right, you can see a picture of what the synthesizer looks like. It's a very small benchtop unit, and it runs completely automated 24/7. So that's followed by our directed high specificity DNA assembly for the next part of the process. So what this is a deterministic assembly of oligos, which avoids errors typically observed with conventional PCA. Traditionally, oligonucleotides are assembled into double-stranded DNA using polymerized cycling assembly, PCA, in which pools of overlapping oligos are thermally cycled in the presence of a DNA polymerase, and you have complementary oligos anneal and extend to form longer fragments, which are subsequently amplified by PCR. PCA, however, is inherently susceptible to hybridization errors, as oligos can misanneal to partially complementary sequences, which is a risk that increases with sequence length and complexity. To address this limitation, we have developed this directed high specificity DNA assembly method, one part of which is that it reduces the oligo pool complexity per reaction without sacrificing throughput or yield. And what this does is it significantly lowers the risk of mishybridization, particularly for highly complex sequences. And with this approach, we are able to successfully build fragments that cannot be made by traditional PCA. So if we look at things like low and high GC content, long repeats, hairpin structures, ITRs, inverted terminal repeats, homopolymers, and other difficult motifs can be successfully handled with this method. So that was the second part of Elegen's Gen 2 cell-free DNA synthesis platform. And that's followed with the final and third step, which is our cell-free cloning work cell. After assembling the oligos into double-stranded DNA, we then move these constructs into our virtual cloning process. So here, both the DNA assembly and the virtual cloning run on fully automated work cells, which are built from standard laboratory equipment, including liquid handlers, thermocyclers, and related instrumentation. So using a compact set of these work cells occupying less than 100 square feet of lab space, we clone thousands of genes each week, including constructs up to 7 kb in length. And throughout this process, we apply long-read sequencing to verify both the purity and sequence accuracy of the piece of DNA that's being constructed. As we'll explain in the next slide, we use a proprietary and selective amplification step that enriches only sequence-specific molecules. This ensures that every gene we ship contains over 95% sequence-specific DNA. We mentioned error rate, so how do we calculate error rate? So to ensure the highest quality, you must be able to distinguish true DNA synthesis errors from background sequencing noise. So we do this by measuring the synthesis error rate using UMIs, or unique molecular identifiers, across a broad set of custom sequences. So what this process looks like is we have, in part A of the figure, we have a pool of DNA, and it may have some synthesis errors in it. So then we take those UMIs and we tag those to the end of those pieces of DNA. That's shown in figure B. So now we have each molecule has its own, essentially its own barcode. We then take that pool of DNA amplify it up. So we have a few copies of each one of these individual barcodes. And then we put that through NGS sequencing where we get a certain read depth. So we have multiple reads of each of these individual pieces of DNA. And then if we look in figure D, or part D of the figure, we can see how we actually use that information to call out a synthesis error as opposed to a sequencing error. So in the top part of figure D, what we have is some read scores along the piece of DNA. We have gray and red sections. So the gray would be a consensus sequence where we have everything agreeing, and we have a few small red bars where we have maybe different calls on different positions of this piece of DNA. And those we can attribute to the sequencing error because they're very small and very diverse across the piece of DNA. But then if we look at the bottom part of figure DNA, at this position A, fourth from the end, what we have is a consensus, red flag. So this is where all the different molecules across the different, barcodes will all agree that there's a true then synthesis error in that position. With this, very automated and detailed look at the piece of DNA, this allows us to have a reliable calculation of a median error rate at 1 in 70,000 base pairs, and enables us to ensure that we have a very high quality metric for each piece of DNA that is shipped to the customers. And then finally, another key part of how Elegen is able to reliably and consistently produce high quality DNA is that we have a DNA synthesis guided by a machine learning platform or algorithm. Our entire build process, from receiving sequence submissions to fabrication to QC testing, is guided by machine learning. So every sequence submitted to our portal is immediately analyzed in terms of synthesis complexity and given a score. And then based on this scoring, the ML platform decides which path to take at key points in this process. So as an example, we can say if the machine learning algorithm finds a problematic sequence, it will say, okay, this sequence should be constructed with a few different parallel pathways. So that's kind of shown in the middle figure, where this piece of DNA takes multiple different routes through our manufacturing process. So this will increase the likelihood of success at that particular steps because we essentially have more chances to get this right. So doing this dynamically ensures that we spend only the time and cost where we need to, but it also ensures that we can have success when there may be more risk. And due to the inherent automation and parallelization of our process, the cost to doing this for Elegen is essentially very small to none. So over time, this improves both the efficiency and the overall reliability of our process, and is one of the key components in being able to deliver high quality DNA in a reliable and consistent manner to our customers. Now I will turn it over to Brittany Ensman, who is our Senior Product Manager.
Thanks, Michael. In this part of the webinar, I'll be talking about the sustainability of our Gen 2 system, as well as the applications for Infinia DNA and the savings that we translate to our customers. Gen 2 was designed from the ground up to make DNA production more efficient and sustainable. The entire platform is fully automated and supported by just 2 engineers. Unlike large factories that cultivate bacteria for conventional cloning, the Gen 2 platform is extremely compact and requires very little infrastructure. The oligo synthesizer occupies just 12 square feet, and that each work cell takes up only about 45 square feet. The small footprint results in minimal operating overhead and enables low-cost scalability. So that means if capacity is expanded, it's simply by adding work cells and not more buildings. The result is a major productivity advantage. We can make over 10 times more genes per square foot per month compared to a conventional factory. And as a bonus, we also use fewer reagents, we produce less waste, and use less energy to make the DNA. Now let's look at some of the applications for which Infinia DNA is used. Starting on the left with mRNA vaccines, Infinia IVT-ready DNA has an encoded poly-A tail and a fast turnaround, which is ideal for vaccine development. Because we deliver the templates quickly, researchers can rapidly screen and optimize leads and move mRNA candidates forward. This is especially important for response to epidemics like we saw with COVID-19. The speed is also critical for personalized cancer vaccines. Infinia's fast turnaround allows for the rapid generation of patient-specific sequences that are essential for tailored oncology treatments. Moving to the center, we support genome editing as well as cell and gene therapies. Whether you're utilizing CRISPR systems for gene editing or viral vectors for gene transfer, we provide high-fidelity DNA required to develop these advanced modalities. And for the right, Infinia DNA is used to develop antibodies and therapeutic proteins. Our DNA serves as a robust template for consistent in vitro and in vivo expression. Currently, we provide RUO DNA, though the platform is amenable to scale-up and GMP manufacturing through our partners, ensuring a seamless transition from the bench to clinical application. Now let's focus on one specific application, prophylactic vaccines, to illustrate some of the benefits of Infinia DNA. When developing a prophylactic vaccine, the process starts by leveraging AI and computational tools to design variants of the target antigen that maximize stability and potency. The next step is to synthesize DNA-based IVT templates so that mRNA can be made in vitro and functionally assessed. However, at this stage, developers often encounter a DNA bottleneck. On the right-hand side of the diagram in red, you will see the traditional path of using a plasmid-based template. These can take over a month to source and require significant effort to build internally. They also carry risks of endotoxins and have design constraints, especially if sequences are cytotoxic to E. coli. On the left in green is the Infinia path. Our IVT-ready DNA has only a 2-week lead time, enabling you to receive templates and start functional assessments immediately. Because Infinia is cell-free, there are no cell contaminants or restrictions for toxic sequences, allowing for a broader design space where you can test your best candidates and not just the clonable ones. The poly-A tail is already encoded, so you can move straight to producing mRNA for testing. This speed accelerates the design, build, test, learn cycle, allowing you to optimize variants and advance lead candidates to clinical trials faster. So ultimately, Infinia DNA ensures that life-saving vaccines reach patients with unprecedented speed. Now here I want to show you some performance data on our IVT-ready DNA compared to plasmids from an external vendor. So here we compared 2 reporter genes, mCherry and firefly luciferase or FLuc, and 2 tail lengths per gene, so A90 and A130. Both the IVT-ready DNA and the plasmids had the poly-A tail tracks encoded in the DNA templates. On the left, we show the mRNA peak length and dispersion for both genes. mCherry is in red and FLuc in blue for each tail length. The mRNA derived from the IVT-ready DNA is on the bottom 2 panels, and the plasmid DNA is on the top 2 panels. The templates with the A90 tail are on the left, and those with the A130 tail are on the right. As you can see, when comparing the peaks across plasmid and IVT-ready DNA formats, they're really similar, indicating similar mRNA size and integrity. On the right side, we have gone a step further and compared the functionality of the FLuc protein from IVT-ready DNA and plasmid DNA. The luminescence on the x-axis indicates that the mRNA was successfully translated into a stable functional protein. As you can see, both formats are comparable in this respect. Now that we have shown that Infinia IVT-ready DNA and plasmid DNA have similar performance, it's important to note that the former was produced 20 to 77% faster than plasmids. So for the same performance, you essentially get Infinia DNA much faster. Now, how much does Infinia DNA cost? We're happy to share that Elegen recently reduced prices across the board by 50%, making Infinia DNA more accessible. To put that in context, let's look at a concrete example. So consider a 4.5 kb Cas9 sequence, which encodes the nuclease that is commonly used for CRISPR editing. When you compare the costs, Infinia linear DNA is about 22% less expensive than a major DNA fragment vendor for the same construct. Importantly, the linear DNA has a 10-fold lower error rate than the fragment vendor, and every construct is sequence verified. So note that vendors of fragments typically do not verify their DNA. Now, what if one wants a Cas9 construct and a plasmid instead? Well, you still save with Infinia, as plasmid DNA, the price is about 27 to 54% lower than other vendors. And for those looking for Cas9 in an IVT-ready DNA format, our product is approximately 9% less expensive than a similar competitor offering. Importantly, these savings don't come at the expense of quality. Every construct is NGS verified, so customers get both efficiency and high confidence in the accuracy of their sequence. I'll close with 3 takeaways on how cell-free DNA is shaping the future of genetic medicine. First, it removes the constraints of bacteria. Without relying on cells, researchers gain a much broader design space and can test more variants without the limitations imposed by cloning. Second, it changes the economics of DNA production. With a smaller footprint, lower capital requirements, and higher efficiency, DNA can be made faster and at lower cost, delivering immediate benefits to researchers. And finally, together these advantages help break the DNA bottleneck. Instead of waiting weeks for conventional plasmid templates, researchers can move quickly, accelerating the path to discovery to clinic. And with that, I thank you for listening to our webinar today and invite you to reach out to us or visit our website at elegenbio.com. And we are happy to take any questions.
Thank you both. Excellent presentations. And thank you to the audience for submitting so many great questions. Feel free to continue adding questions as you think of them, and we will jump right in. First question is directed to you, Michael. Can Elegen synthesize sequences that are toxic to cells?
Yes, thank you. So because Elegen's production platform is completely cell-free, we can easily produce sequences that are typically toxic to host living cells.
All right, thank you. Another one for you. What kinds of complex DNA sequences can Elegen build?
Yeah, thanks. So Elegen's manufacturing process can successfully build things like high and low GC content, long repeats, hairpins, ITRs, and homopolymers. So the sequence complexity thresholds are all available on our website, and we have a knowledge base with acceptance criteria.
Thank you. Brittany, I'll toss one over to you. How does IVT-ready DNA compare to plasmids?
Oh, okay. Thank you for that question. Because Elegen's manufacturing process doesn't involve cell-based cloning, we found that IVT-ready DNA templates can be produced up to 77% faster than conventional plasmids and generate mRNA that's comparable in size, integrity, and functionality. We actually have several application notes and white papers with data on our website, so we invite you to take a look.
Thank you. Keep going with you. We've had a couple questions come in regarding pricing. How does Elegen's pricing compare to other vendors?
Yeah, great. So we recently lowered our prices by 50%. So we now offer linear DNA starting at 7 cents a base pair. Our IVT-ready DNA is also starting at 7 cents a base pair, but we also have a tail fee on top of that. And then lastly, our plasmid DNA starts at 12 cents a base pair. But if you find a price that's lower from any other vendor, you just tell us and we will match it.
Awesome. Michael, let's go back to you. What quality control methods do you use?
Yeah, thanks. So Elegen has 3 core QC methods, which are used for all our Infinia products. So the first is that we verify yield via high-sensitivity fluorescence spectrophotometry assay. Secondly, we use long-read sequencing to confirm the sequence ID. So this ensures that we have a 100% match with no things such as SNV in your sample. And then finally, we use the long-read sequencing to assess the purity of the sample and verify that there are minimal side products present. So importantly, a sequence will only ship if it passes all 3 of these criteria, and you get a pass-fail status for each test upon shipment.
All right, and then how do they order Elegen's DNA?
Yeah, so that's simple. You can just go to our online portal. You can set up an account, select a product, upload your sequences, and we have a great team to assist you if you have any questions.
Thank you so much. Let's see. Why do you use long-read sequencing in your QC?
I'll take that one. So we use long-read sequencing to accurately verify complex DNA features. So this can include repeats, hairpins, high GC content. These things can be really difficult to resolve with short-read methods. And because long-read sequencing reads constructs really from end to end, it confirms that all of the sequence elements are correctly assembled and positioned within the final product.
Thank you, Brittany. Another one to you. Can I order DNA in plates or tubes?
Oh yeah, so we currently offer two plate options. We can ship in a 96 well microplate or a 384 well eco qualified microplate, but currently we do not ship in tubes.
Okay, good to know. Michael, can I order the DNA normalized?
Yes, this is possible. So we offer a couple different formats. So the DNA can be lyophilized, dissolved in water, or dissolved in TE buffer. So go ahead and contact our customer support for the normalization option to help get that organized for your order.
All right, I'm trying to not say any duplicates here. Michael, are you planning to launch any new products soon?
Yes, so we are continuously developing our Gen 2 platform, and we do intend to release new products and new features in the future. So our portal, our website will have all the new releases, and also follow us on LinkedIn for the best updates and the latest info.
Amazing. Well, thank you both so much. And thank you to our audience for your active engagement. If we were not able to answer your specific question, we will do our best to follow up after the webinar. As a reminder, a recording of this session will be available on demand within 24 hours. Thank you for joining us today, and we look forward to seeing you next time.